So, mankind have reached the Earth and the Moon repeatedly, a distance of over 300,000 kilometers above sea level, and have done so 6 times already. Almost 20 men have been there. But until recently, there was only a sole attempt to reach the deepest point of the ocean, a mere distance of only 11 kilometers below the sea level. And that explains how difficult it is to reach deep ocean. And only last year, James Cameron, with his deep sea Challenger, have made it again to the Mariana Trench, to a total of 2 missions and only 3 men. I believe I do not need to introduce the HPNS signs and symptoms to this audience again. But we do need to think about the fact that these signs and symptoms are attributed to some kind of neurological malfunction. And as a reminder, synaptic transmission happens when first an action potential invades the presynaptic terminal, causing the opening of voltage-dependent calcium channels. Calcium ions then flow into the synaptic terminal, causing vesicle fusion with the membrane, leading to neurotransmitter release into the synaptic cleft. The neurotransmitters then bind to the postsynaptic receptors, causing the receptors to open, allowing ions to flow into the postsynaptic terminal, causing depolarization. So in essential, it is a chemical process designed to deliver an electrical pulse from the presynaptic side to the postsynaptic side, from an action potential to the depolarization, postsynaptic depolarization. And it has been shown in the past that synaptic transmission in general is suppressed, and more specifically that there is a reduction in vesicle fusion and depression on neurotransmitter release. Some of these works have been presented here today. That might point out that an earlier process in the synaptic transmission is also affected by pressure, namely the entry of calcium into the terminal, and indeed a few works in the past have suggested that, but have done so, a few works including Grossman's of course, and but they have done so in an indirect manner. Thus, it remains the question, what exactly is the pressure effect on the activity of these channels? And this was the goal of our research, to directly measure the currents through the channels while under hyperbaric pressure. These channels are built out of an alpha-1 subunit, the main subunit which holds the ion conduction pore, the voltage sensor, and the channel gating area, and of a beta, alpha-2 delta, and gamma subunits, which mainly have modulatory and synergistic properties. They have 2 major families, the high and the low voltage activated, and they are subdivided by pharmacological blockers and bodily distribution. The main change between the different types is the alpha-1 subunit, and until today there are at least 10 different isoforms that each one defines the specific type of calcium channel. These voltage-dependent calcium channels are expressed throughout the neuron, from the dendrite, proximal dendrite, soma, and the synaptic terminal. And in each one of these regions, different research have shown their expression in these regions. Our research first focused on the dendritic and somatic channels, such as the L and the T. To do so, as Amir Moore have explained before, we injected a mixed RNA into xenopus laevis oocytes, which is just a fancy name for saying frog eggs. And essentially, what this RNA does is encoding for the specific channel of interest. And of course, it doesn't express one, but many of them on the entire surrounding of the membrane. The oocytes are about 1 millimeter size, which is considered to be big in electrophysiology. We can then poke them with 2 electrodes and perform a 2-electrode voltage clamp inside a pressure chamber, which you already saw, compressed by helium due to its inert properties. My research was done up to 50 atmospheres, although this chamber can hold up to 400 atmospheres. The two-electrode voltage clamp actually allows us to measure for each depolarization of the membrane a specific current, and then we can sum up this data to construct an IV curve, which tells us what is the size of currents going through the channel in relation to the size of depolarization. So we move on to the results. The first channel was the T. As you can see here in black, 1 atmosphere, red and blue, 5 and 50 atmospheres, a considerable reduction of the currents. If we look at the maximal currents in the IV curve, we can build this figure, which tells us that currents reduce as pressure increases. Decompression did not succeed to recover any of the currents. Thus, we can say that for the T-type, pressure suppresses the maximal currents. Both the threshold voltage and the voltage which generated the maximal currents in the channel did not change at pressure. Thus, we can say that there is no apparent change in the voltage sensing of the channel. It is interesting to note that at about 1 megapascal, which is the pressure of 100 meters of seawater, which is the depth in which divers begin to complain about the HPNS sinus syndromes, the effect of pressure on the channel has already reached its maximal capacity, which might suggest that this channel is indeed involved in the HPNS mechanism. The channel is expressed in heart and brain rhythmic centers, and smaller currents through the channel could mean slower rate of these centers. It is expressed in the reticulothalamic nucleus, and slowing it down could mean changes in EEG. And that exactly was demonstrated by Grossman et al. On the work done on the French Navy divers. You can see a shift from the alpha to theta wavelength waves, meaning slowing down of EEG. The channel is also expressed in the thalamus, responsible over sleep, awareness, and activity, and disturbance in its activity could lead, on one hand, to sleep disorders, and on the other, to drowsiness. The channel is also expressed in the striatum, that could lead to reduced cognitive performances. The striatum is also responsible over movement planning and modulation, and that could lead to impaired coordination. And in the cortex, responsible over sensory inputs, and that could lead to vision and auditory disturbances. Now, it happens to be that this constellation of sinus symptoms is actually part of the HPNS, and which is why we suspect it does have a role in its mechanism. The channel is also expressed in the SA node, the cardiac pacemaker, and of course, slowing it down would lead to bradycardia, and this was also demonstrated in an isolated rat heart, as you can see here, slowing down of the pace at 60 atmospheres. The next channel we moved to was the L type, and as you can see here, black is 1 atmosphere, red and blue 5 and 50. The currents were augmented at pressure. This figure on the right also demonstrates an increase of the currents as pressure increases, and decompression only partially succeeded to recover the currents. Thus, we can say that for the L type, pressure augments the maximal currents. Again, there was no shift in the voltage sensing of the channel. So for the first time, we saw a pressure selectivity on affecting voltage-dependent calcium channels. And it is interesting to note that few works in the past have not only suggested slowing down the heart pace, but also an increase in the contractility force of the cardiac muscle. And since this channel is expressed in the cardiac muscle, that could be an explanation for this phenomena. Stronger currents through the channel, stronger conductivity force. These results in the L-type could also explain or correlate with previous results done in our lab, as I showed in earlier today, by work done by Tal Palau, that could explain the increased transfer function in a given neuron, where a reduction in the field excitatory postsynaptic potential was witnessed, but the output of the population spike has remained unchanged or only slightly reduced, And a possible explanation could be dendritic or somatic boosting of the currents in between these two regions. And since the L-type calcium channel is indeed expressed in the proximal dendrite area, that could be a possible explanation. So I remind you that our goal was to directly measure current through the channel, and we have found a selectivity of the pressure effect already on these two channels. Here, sum up of compression to 25 atmospheres. We then move on to check, to examine channels expressed in more synaptic areas, such as the N and the PQ. For the PQ, as you can see in this IV curve, compression to 25 atmospheres did not significantly change the currents. This is an example from one ore site. Thus, we can say that for the PQ, pressure does not affect the maximal currents. Again, there was no shift in the voltage sensing of the channel. So we can say that the PQ channel may be insensitive to high pressure, and that correlates with results from Etzion and Grossman experiments, which were just shown here, where in Pourquinier fibers, compression to 100 atmospheres did not significantly change the calcium spikes, as you can see here. And in Pourquinier fibers, it is important to know that the PQ channel is the most dominant one. Various works in our lab throughout the years were done on the relationship between calcium concentration and high pressure. This work by Grossman and Kendig, which I showed earlier today, compression to 100 atmospheres reduced the response to stimulus in lobster excitatory junctional potentials, and it was mimicked by reduction of the calcium concentration. The same happened when potentiation was measured. Again, compression to 100 atmospheres and reduction of calcium concentration by half. Thus, we can say that reduced calcium concentration can mimic the high pressure effect. On another work done on frog, which is yet to be published but already submitted, you can follow the black line in 1 atmosphere. This represents calcium currents. In red, 70 atmospheres, reduction, significant reduction of the calcium currents. But in green, when at 70 atmospheres, the calcium concentration was elevated by 5-folds here, there was at least some recovery of the calcium currents. Thus, we can say that elevated calcium concentration can antagonize the high pressure effect. A similar thing was demonstrated by Talpala, which just showed this work before this presentation. And in short, in blue, 1 atmosphere and regular calcium concentration, red, 100 atmospheres, a significant reduction of the field excitatory postsynaptic potential, as you can see also in the figure on the right. But when under 100 atmospheres, the extracellular calcium was increased or doubled to 4 millimolars, at least some recovery was witnessed. Now, if we remember that in each synapse there are at least there are more than one type of voltage-dependent calcium channels, we can postulate that this partial recovery may occur through synaptic release sites that are close to voltage-dependent calcium channels that are less sensitive to high pressure, such as the PQ. The last channel we moved to, and this work was done very lately, the N-type, which is the classical neuronal channel. In black, 1 atmosphere. In red, 3. Blue, 25. And green, 50 atmospheres. We witnessed an augmentation of the currents, and this is not expected. We can say that for the N-type, pressure augments maximal current. You can also see on the figure on the right. And decompression only partially recovered the currents to control levels. Again, there was no shift in the voltage sensing of the channel. Since these results are somewhat surprising, we now guess, we don't have yet the explanation for that, But we think that may be because we either we express this channel from 3 different organisms. The subunits came from rat, rabbit, and human. And it might be that this chimera doesn't really mimic the mammalian behavior. That's one explanation. Another one is that in order to express this channel in oocytes, we only need to express 3 subunits out of the 5. And that's what we did in all of those channels. And it might be that a subunit that was left out that is not crucial for the expression and functionality of the channel is pressure sensitive in another way, or pressure sensitive in a way that will differently affect this channel at pressure. So to sum up what I've just shown you, pressure does not seem to affect the channel voltage sensing. Reduction in the T-type current may be involved in many HPNS signs and symptoms. Augmentation of L-type currents may explain increased transfer function in neurons and increased contractility force in cardiac muscle. Insensitivity of the PQ channel may contribute to partial recovery of field excitatory postsynaptic potentials induced by elevated calcium concentration. And high pressure, of course, selectively affects different voltage-dependent calcium channel types. And again, if we remember, this is a sum up of compression to 25 atmospheres. We also have 10 and 50 and 5 atmospheres. If we remember that each synapse has more than one type of voltage-dependent calcium channel, I believe the importance of this work is that if we would know the channel type distribution and combination in a given synapse, we may determine its behavior under pressure. And this is what we tried to do in a review published 2 years ago, to give the postulated high pressure effect on a network, and a neural network, from the input signal through synaptic transmission, signal integration, signal transfer, and the output signal. And you can see that we concluded that the output signal would depend on the specific type of density of expression of these channels. So we do have a master's student, Alice, as Amir just stated, but being the last PhD student, I believe, probably, I'm feeling obliged to talk a bit about the future. We do want to find out the reason for the pressure selectivity. We can do that by scanning for sequence and structural differences, and that can be done either between different channel types, sensitive and insensitive ones, or between different animal species. An example for this, such a work was done by Daniels, the chair for this panel, on a pilot whale, on a glycine receptor of a pilot whale, where a few differences in the sequence were found between the whale and human and rat, one of them being in the ligand binding site of the receptor, and he demonstrated an increased affinity, a left shift in the dose curve. And it could be that this increased affinity of glycine can explain a protective effect against hyperexcitability, allowing this mammal to go to extreme depths. Another option to go about is to create channel mutations. And for some of you, this is the first visit to Israel. We are in the very south point, and I came over here from, actually where I grew up is a small village called Ramot, sitting next to the Sea of Galilee, where Jesus walked on water. And having that on the background, I would like to thank Professor Grossman, which you can see here diving in the Red Sea, Dr. Amir Moore, diving here in the Mexican cenotes. A special thank to Merav Moore, which greatly helped me with expressing these channels in the oocytes, and also agreed to marry me. And just a year ago, we had a child born. And of course, thank you for listening.
Thank you, Ben.